Hello, my name is Joshua Mugabe. I welcome you to Cells and Tissues. This is a thin slice cut vertically through the tip of the plant stem as seen under a microscope. So we are looking at a section of the plant stem that has been cut through vertically, and it is seen under a microscope. So what we are seeing here, this is a stem, and this part here is the growing point. Growing point, what we would call like a bud, terminal bud. And here we have a bud which can be seen here. Here we have the leaf. This is the leaf. And in between the leaf and the stem, we have the bud. Also here the bud, and this is the leaf. But you can see that they are small squares. Those small cubicles that you are seeing, those are cells. The slice is so thin that light can pass through it. So when they are cutting the slice, they make it very thin so that when it is put on the microscope, light can easily pass through it. The stem leaves and the buds are made up of large, tiny box-like structures, and these are called cells. So when you look at the stem, the whole stem, you look at the leaves, you look at the buds, the growing points, you will see that they are made up of large number of tiny box-like structures, and these structures that we are seeing, they are the cells. The slice of the tissue must only be around 0.01 to 0.1, that is in millimeters thick, if cells are to be seen clearly. So when we are slicing the tissue, for us to be able to see those cells clearly, we should make sure that the size, the thickness of the tissue is between 0.01 to 0.1 millimeters. That is when we can be able to see clearly the content of the tissue. Thin slices of plant tissues can be made by hand using a sharp razor blade. That one can be made by using a sharp razor blade. We can cut it using the hands. The slices are called sections. When we are slicing, we call them sections. When you cut from top to down, that one becomes longitudinal section. When you cut across, that one becomes transverse section. So the slices are made in sections. So the cutting sections, we have two sections that we can cut. We have the transverse section. This one here, we are cutting using a sharp razor blade, cutting across. That one becomes transverse section. Then we have the longitudinal section. Longitudinal section, we are cutting along the length. That one becomes longitudinal section. The slice of tissue must only be 0.01 to 0.1, that is in millimeter, that is the thickness, if we want to see the cells clearly. The sections of the plant tissue can be cut by a hand. We can use a razor blade, like the way we are seeing here. We have our razor blade and we are cutting using the hand. Soft tissues, particularly animal tissues, they need to be embedded in wax or plastic, and the sections cut by special apparatus. So when we are having our tissue, we say that it is a soft tissue, like animal tissue, it becomes very hard to cut. So for us to cut it, we have to embed it in wax or plastic so that it becomes hard, and when we are cutting the sections, we can use special apparatus. Remember, we said we want this thickness to be 0.01 to around 0.1 millimeter thick. So we need to make the soft tissue a little bit hard so that we can easily cut that thickness that we want. So to make that one, we make sure that we embed it in wax to make it a little bit stronger so that we can cut it that thickness. Extremely thin sections can be cut by this method. So those thin sections that we want to cut, we can use these methods of embedding the substance in wax. Then we can cut that small thickness of a section. A group of cells from animal tissue. We want to look at a group of cells from animal tissue. All cells, they consist of a cytoplasm, they have a nucleus, and they have a cell membrane. So when we look at our cells here, you can see that this cell has a cytoplasm, the liquid or semi-liquid part of the cell. It has got a nucleus in the middle, approximately, and it has got a cell membrane, the one covering around the cell. That is the animal cell. The cytoplasm is a living semi-liquid material. So what we call the cytoplasm, this one inside the cell, is a semi-liquid living material. It contains many structures which are not visible at this magnification. So because the magnification is low, we can see the cytoplasm, but we cannot see the structures that are in the cytoplasm because the magnification is small. Therefore, the organelles or cell organelles are not visible because of the low magnification. But the cytoplasm contains very many structures which are not visible at a low magnification. All the processes which keep the cell alive, they take place in the cytoplasm or the nucleus. So there are very many processes that occur in the living organism: respiration, excretion, all those ones, they take place in the cytoplasm. And some of them, they will take place in the nucleus, but most of them, they take place in the cytoplasm. All those processes that keep the cell alive are taking place in this semi-liquid material. The nucleus is a specialized region of the cytoplasm, and this one regulates all the chemical changes that take place in the cytoplasm. We have the nucleus, which is in the cytoplasm, and this nucleus is a region in the cytoplasm and in the cell which is specialized. It regulates all the chemical changes that take place in the cytoplasm. We have said that all the reactions, most of the reactions take place in the cytoplasm, and those reactions are regulated by the nucleus. It sends chemical signals to the cytoplasm, which starts or stops the chemical process going on in it. So it is the nucleus which gives signals or sends information, messages to the cytoplasm for a particular reaction to start, and it is also the one which gives information for the particular reaction to stop. So the nucleus is the controller, it is the regulator of all the chemical processes that are going on in the cytoplasm. The nucleus also controls cell division. In the nucleus, we have genetic materials, and those genetic materials are the ones that are responsible for cell division. It is the nucleus that controls a cell to divide into many cells, what we call cell division. That one is controlled by the nucleus. The nucleus contains a substance called DNA. That substance called DNA or what we call deoxyribonucleic acid, it is found in the nucleus, and the DNA controls what kind of cells are going to be produced. For example, they can be liver cells, muscle cells, or blood cells. It is the DNA in the nucleus which is going to control which type of cells should be produced. Like we have said, it is the nucleus that controls cell division. So when a cell is dividing and producing other cells, now which cells are they producing? Now it is the DNA in the nucleus which is going to control the type of cells which are going to be produced during cell division, whether they should be the liver cells, the muscle cells, or the blood cells. Chemicals produced by DNA, they escape from the nucleus and regulate all the chemical reactions in the cytoplasm. So, in the nucleus, we have a substance called DNA. And this substance DNA, we are saying it is the one that determines or controls which type of cells are going to be produced. And the DNA, the chemical produced by this DNA is going to escape from the nucleus and enter into the cytoplasm. When it enters into the cytoplasm, it is now going to regulate all the chemical reactions that are taking place in the cytoplasm. Cells without a nucleus cannot divide. If a cell has no nucleus, it cannot divide. If a cell does not have a nucleus, it does not undergo cell division. We have cells that we know that do not have a nucleus, like the red blood cells. So red blood cells do not undergo cell division because they do not have a nucleus. The cell membrane encloses the cytoplasm and stops it flowing out. So we have our cell membrane. The cell membrane is that part covering the cytoplasm or enclosing the cytoplasm. It is our cell membrane. And the cell membrane prevents the cytoplasm from going out of the cell. That is the work of the cell membrane. The cell needs to take up oxygen and food substances to keep it alive. So the cell, of course, has to be alive. And being alive, it will need to take in oxygen, and it needs to take in food materials like glucose, amino acids, and so on. The cell membrane allows these substances to pass into the cytoplasm, but keep unwanted and possibly harmful substances out. So it is this cell membrane that is going to control what is entering and what is moving out. There are some substances which are going to be prevented from entering the cell by the cell membrane. It will reject them. And then there are other substances the cell membrane is going to allow to enter into the cell. And these substances, we have talked about them, like for example, oxygen and food substances. So it is the cell membrane which allows these substances to pass through into the cytoplasm or to keep them out of the cytoplasm. They may be harmful or they may be not wanted into the cell. It is only about 0.00001 millimeter in thickness. So the cell membrane is very thin. It is very thin so that it can easily allow materials to diffuse. About 0.00001 millimeters. That is the thickness of the cell membrane. If it is damaged, then the cell will die. So if you have your cell membrane damaged, if it is damaged, then the cell is going to die. These cells have been scrapped from inside the cheek. The cheek is inside the mouth. And when you scrap those cells from the cheek and you magnify them, this is what you are going to see. You will see the cells of the cheek. They have the cytoplasm, they have the nucleus, and you will be able to see the cell membrane. That is when you put them under a microscope. They are not stained, but viewed with special lighting. The ones that we are seeing here, they have not been stained, but they are seen under a special lighting. The magnification is 950. These ones have been magnified 950 times for them to be as big as the way you see them. Plant cells. We want now to look at the plant cells. The cell membrane is pressed against the inside of the cell wall. By the pressure, of the virtue and is difficult to see. We have the cell membrane. The cell membrane is this small layer. It is this small. It is really placed very tightly on the cell wall. What you are seeing, this line here, this line that you are seeing is the cell wall. This line here is the cell wall. Now the cell wall, we can say that one, the white tube, is our cell wall. So we can say inside it there is a black line. That black line inside, next to the cell wall, we can assume it is a combination of the cell wall interior and the cell membrane pressed against the cell wall. And what is pressing it is the virtue. The virtue exerts the pressure, and when the virtue exerts this pressure to the cytoplasm, you can see the cytoplasm now will exert pressure onto the cell membrane, and then the cell membrane will be pressed against the cell wall. So this one is our virtue, and this layer that you are seeing here in between the cell wall and the virtue, that is the cytoplasm. So this virtue is exerting pressure on the cytoplasm, and then the cytoplasm will transfer that pressure to the cell membrane, which is going to be pressed against the cell wall. So it is very difficult for it to be seen because it is pressed onto the cell wall. The cell wall is made up of cellulose. So we have seen our cell wall here, and this cell wall that we are seeing out of the cell, that cell wall is made up of cellulose. It is not made up of any other material, but it is made up of cellulose. It is permeable. That is, it lets water and dissolve these substances to pass in and out freely. So our cell membrane is permeable. It can allow substances to enter. At the same time, it can allow substances to go out of the cytoplasm because it is permeable. It is going to allow the water will enter, and then we have things like nutrients will enter, oxygen will enter, and it will allow other materials to get out, like waste material, nitrogenous wastes, and so on, to move out of the cell freely. The virtue, this one contains solutions of chemicals such as salts, sugars, and sometimes colored substances. That is the virtue. The virtue is this middle part here. This middle part is what we call the virtue. And this middle part contains solutions of chemical substances. And these chemical substances, they include salts, they include sugars, and sometimes colored substances. are the ones that we find in this vacuole, which is in the middle of the cell, in the middle of the cytoplasm. The solutions is called cell sap. So our solution that we are finding in the vacuole is called the cell sap. It is called the cell sap. That is the solution which we find in the vacuole. Its outward pressure on the cell wall keeps the cell firm. So we have looked at our cell wall and we have seen it has a firm structure. And that firm structure that you see is a result of the pressure that is coming from the vacuole. The vacuole is applying pressure to the cell wall, and when it applies pressure to the cell wall, that pressure keeps the cell firm or rigid. These cells are from a moss leaf, and the magnification is 400. So we have our cells here. They have been extracted from a moss leaf. We can see the vacuole in the middle. We can see the chloroplasts. Those substances that you're seeing, those are chloroplasts. And you can see the cell wall. You can see the cell wall. Chloroplasts, these ones contain a green pigment, which is chlorophyll. And this one, it absorbs sunlight, which helps the plant to make food. So we have the chloroplasts. These chloroplasts, they contain chlorophyll, and that chlorophyll is the one that is used in the manufacture of food for the plant. This green substance is called chlorophyll, and it is found in structures called chloroplasts, and its function is to absorb sunlight energy to carry out photosynthesis. Cells from green plants or green parts of plants, such as the leaves, they contain chloroplasts. So when you get different cells of different parts of the plants, they are green parts on the plant, like the leaves. Those green parts of the plants, they contain chloroplasts. And what is making them to look green is because of the presence of chlorophyll. The chloroplasts are always in the cytoplasm. So when you look at our cells, we have said that this one is the cell wall. That one is the cell wall, which is enclosing the cytoplasm. And you can see that our chloroplasts are in the cytoplasm. You find them in the cytoplasm. They are embedded in the cytoplasm. Those are chloroplasts. All cells, they have a cytoplasm. They have a nucleus and they have a cell membrane. That is all cells, if they are animal cells and plant cells. They have a cytoplasm, they have a nucleus, and they have a cell wall. Plant cells also have a cell wall, a central vacuole, and in many cases, chloroplasts. That is now for the plant cells. They have got a cell wall, they have a central vacuole, and in most cases they have chloroplasts, meaning that these organelles or these parts are not in animal cells, but we can find them in plant cells. There are exceptionals of this general statement, like the red blood cells. For example, for them they don't have a nucleus. We have said up that all cells, they have got a nucleus. But there are some exceptions, like the red blood cells. For them, they don't have a nucleus. Animal cells often have vacuoles, but these are small and may be scattered throughout the cytoplasm. So we were saying that the central vacuole is found in plants. But here we are saying that the animals may also have some vacuoles. But these vacuoles are very small and they are scattered throughout the cytoplasm. Whereas when you look at the plants, the vacuole is centrally located inside the cell. Let's look at the stomach. The stomach is an organ and part of the digestive system. So when you look at our stomach here, it is an organ which is part of the digestive system. But if we cut a smaller piece, you cut a smaller piece from the organ of the stomach and you magnify it, this is what you are going to see when you magnify that small part of the stomach. The stomach wall contains muscle tissue. When you look at the stomach wall, this is what you are going to see if you have cut that smaller part. There is the muscle tissue, which is this lower part of the stomach is made up of the muscle tissue. It is made up of the muscle tissue. And even the upper part is also made up of the muscle tissues. So if you get this small part and you cut it, this is what you are going to see. The muscle tissue consists of the muscle cells. Here we have our muscle tissue. This is our muscle tissue. But a tissue is made up of a collection of cells. So when you look at this tissue, you will see that it has got cells, and those are the muscle cells. It has very many cells joined together to form a muscle tissue. So from the organ to tissue and then to cells. Thank you very much for participating in cells and tissues. Please do like, share, and subscribe.